To take, ladies and gentlemen, to proceed to the next presentation with the topic of application of pharmacogenomics clinical annotation tool, which will be presented by the honorable representative from Center for Medical Genomics, Dr. Chakapan Runcharan.
Good afternoon, everyone. For this session, I'm going to talk in the title of Application of Pharmacogenomic Clinical Annotation Tools, or PharmCAT. The aim of this talk is to share you the PharmCAT analysis pipeline that I have used in the CPharm project. The resequencing project of 1,000 Southeast Asia individuals using the 100 pharmacogenes by next generation sequencing panel was established in 2018 under the collaboration of CPharm network. And there are 1,571 samples participated in this project. The sample is from 7 countries of Southeast Asia, together with Greece and UAE. So for this project, the sequencing process was done by Riken, Japan. And the primary sequencing analysis was performed by the Center for Medical Genomics by applying the script which is developed by the Riken, Japan. This slide shows the pipeline, the analysis and the interpretation pipeline that used in this project. It can be divided into two branches. First is discovery, and the second is implementation. PharmCAT is involved in the part of implementation of this pipeline, as it can translate the raw sequence data to the PTX variant interpretation with the guideline recommendation. PharmCAT is an open access, open source, and it has been developing by the team from the Stanford and the University of Pennsylvania. It can help you to extract the pharmacogenomic variants, infer the haplotype and diplotype, also together with provide the genotype-based prescribing recommendation. What do you need to provide for this tool is the user-provided normalized VCF files. So this is show the process of this tool. This tool will creating the genotype, normalized genotype from the input file and matching with the allele definition and combine with the guideline, and then creating the final report. In the final report, there are 11 genes included in the report. And in case of only 10 genes can be reported with the recommendation based on the CPIC guideline. And in case of you want to include the result of the CYP2D6, do you have to analyze with another tool, which is the isolate, and then you can incorporate the result from the isolate into the final report, making the total of 12 genes in the report. So to creating the VCF file, first thing is the FASTQ file will be removed for the low quality base bioinformatic, then align to the reference, which is the HG 38. And then variant calling will be called rely on the GATK. For this work, I used the haplotype caller with a few arguments, including the false active and the disable optimization and the emit all site. Based on these arguments, so the VCF file will look like this. So it includes both variant and non-variant size. And I use this VCF file for further analysis, including Isolate and PharmCAT. So before we running the PharmCAT, there are three things of the general requirement it needs to be concerned first, including the build version must be aligned, as I mentioned, aligned with the HG38. And any position that not included in the VCF is assumed to be no call. And for the chromosome column must be in the format CHR sharps. And also the quality and the filter column are not interpreted. And the VCF file, it should be a single sample. So, and this is the ideal the format for running the PharmCAT, as shown below. Then the next step, I make another script. The aim is to optimize the VCF file and then running isolate and include the result into the PharmCAT report. For more information on this step, so you can check it out at their website or GitHub, how to install and how to running the software. This is show the PharmCAT example of the PharmCAT report. So for PharmCAT report, it can be divided into three sections. The first is genotype summaries. For this section, it will report you the list of the drug based on CPIC guidelines, the genotype and also the phenotype of this sample. And also is linked the information of genotype with the recommendation, as you see in the different color and the marker in front of the drug listed. The cost place here is indicated that this genotype causes the abnormal metabolites, for example, intermediate or poor metabolites, and based on the guideline, the prescription change is recommended. And for the green color, it indicates that you have a normal metabolites and no CPIC guideline recommendation for using this drug. And the second part is the CPIC recommendation will give you the detail how to adjust or how to deal with the dose to treat this kind of genotype. And the third section is the allele matching data. For this session, it will show you the detected allele core related to the allele, the related allele, linked to the related allele. And this is all about the PharmCAT report. The next, I will summary the concordance compare between the PharmCAT and other platform. This slide shows the percentage of concordance between PharmCAT and the GetRM or genetic testing reference material coordination program. You can see that the percent of the concordance between this platform is high. It's quite high in the gene, in the CYP2 to CYP genes, IFNL3 and TPMT genes. And the percentage is quite low in the SLCO1B1 and UGTA1A1. And this is the previous one is from the publication, and also I also created the data from the Thai sample that tests for the allele, star allele calling, and with the compare between the PharmCAT and the Axiom Precision Medicine Diversity Array. The percent concordance is quite the same with the previous one. It's quite high in the CYP2 to CYP gene, IFNL3 and TPMT genes. And SLCO1B1 and UGT1A1, which is low, and based on the that it has been discussing about, because of these two genes is still not well established for the star allele nomenclature, and also the allele definition for this gene is different between different platforms. Summary of this talk: the PharmCAT is an open access. It helps you to extract the genomic variants and also assign haplotype and diplotype, and also including the prescription guideline based on the CPICs. And the PharmCAT result is quite readable and is highly concordant with the GetRM platform. Okay, thank you for your attention. Any question or the comment would be appreciated for further improvement.
Thank you very much. And we do have some questions waiting to be answered here. So I'd like to represent the audience and ask the question right away. The first question, it have the other tools like PharmCAT, and what's the different and or inferior? That's the question.
Can you repeat it?
Any other tool that's similar to PharmCAT? That, to my understanding, I think, yeah. And what's the inferior part? If there is, what is the difference in inferiority? I guess, yeah.
For other software, other tool, rather than PharmCAT to analyze the PCX variant, I know one is a star grasser, but just only analyze for the haplotype and the diplotype, but cannot, but no guideline for that tool.
Thank you very much. And